Movement of molecules in liquid. Remember, the interior of the cell and the exterior of the cell are both fluid. They're both liquids. They're both water-based liquids. Whenever you think about the cell's environment, you're thinking about it being submerged in water or some water-based fluid. Examples of fluids within your body are your cytoplasm and cytosol, which is the liquid inside cells. Specifically, the cytosol is the liquid, the cytoplasm is the space. Let's start with blood plasma. Your blood's liquid, right? Red blood cells, white blood cells, platelets all float in a liquidy serum called plasma. Liquid part of blood. Plasma has dissolved gases, it has a lot of proteins, it has nutrients, it has sugars floating in it. It's not pure water. It got a pH of about 7.4, and it's isotonic with the rest of your body. In fact, the rest of your body's cells are derived from blood plasma. Interstitial fluid is liquid between cells. Lymph is liquid in your lymphatic system. When fluid makes its way over there, it has its own name. And CSF is short for cerebrospinal fluid. So you have blood moving through your body, right? As it emerges, as your blood vessels emerge from your heart, the walls of those vessels are very, 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 very thick. And those blood vessels, or the aorta, the first biggest blood vessel, branches very quickly. Some branches go up to your head, some go to your arms, some go to your gut, some go to your legs. Those branches will branch again, and they'll branch again, and they'll branch again and again and again, until the walls of the blood vessels, and they get thinner every time they branch. So they keep branching until the walls of the blood vessels are only 1 cell thick, and they're fairly leaky. They're so small that you can only fit 1 red blood cell at a time through. And even then, sometimes in order to fit, they have to bend to make their way through. So let's draw some cells that are next door to those super thin blood vessels called capillaries. Just making these boxy for simplicity. I should have given myself more room. Oh well. Okay. So this fluid part of blood is going to be able to leak out and can either be absorbed into the cells to help supplement their cytosol, or it can flow up between cells and bathe, and I'll make this blue just because water, and it stands out blue, and it'll fill the space between cells so that each cell is not just filled with fluid, but it's bathing in fluid as well. This fluid might collect and go drain into veins. Those start off with like a capillary portion of veins, and then they merge to become bigger veins called venules, and then those merge and those merge and those merge until it gets to the biggest, fattest blood vessels called the superior and inferior vena cava that come back to your heart. It does the opposite of what arteries do, where it branches, branches, branches. Instead, veins come from capillaries and merge, merge, merge until you have big fat blood vessels coming into your heart. So that fluid can either go back into your veins, or it can drain through lots of layers of cells and enter parallel passageways to veins called lymph. And lymph is usually drawn as a green, in a green color. So I'll keep with the convention. Those ultimately converge and come back into your veins. You'll learn more about this in your cardiovascular stuff. So don't worry about memorizing the super details of this right now. But know you have blood plasma, that's a liquid part of your blood. That liquid part of your blood is going to help contribute to the liquid portion of cells. They're going to help drink it in when they need it. It's going to surround the space between the cells. And then between the cells, when it fills that space, it's going to need to drain somewhere. You can't just have stagnant water sitting around. And that's going to either drain back into the cardiovascular system as blood plasma again, or it's going to drain out into this separate passageway called lymph. This is where your immune system likes to hang out, as a separate pathway. In a similar way, in your brain, you can make cerebrospinal fluids. You have little capillaries in your brain and special places in your brain. And then there's special cells that can suck the fluid out of your blood without allowing, in an even more controlled way, so that only the liquid comes through. That helps bathe the brain from the inside out. In a sense, interstitial fluid, cytosol, lymph, blood plasma, and cerebrospinal fluid are very close to the same thing. All of them are derived from blood plasma, and all of the cells in your body, save for the very, 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 very most exterior of your epidermis, your skin cells, are bathed in fluid, surrounded by fluid, and fed from the fluid of your bloodstream originally. Now, you already had a lab on this. Excuse me. You already had a lab on diffusion. I gave that to you in hopes of remembering it from introductory biology, but let's recap this here too. I can finish erasing this. If I don't erase it, PowerPoint freaks out. So diffusion refers to the movement of molecules or ions, which are just regular old atoms with a charge, from high concentration to low concentration along a concentration gradient. This process can be passive or it can be mediated by the cell. So passive means it's going to happen by itself with no additional input. As long as there's a passage for it to move, it's going to move. Sometimes, though, the cell will have select pathways that these have to move through, and the cell will control whether or not the molecules move at certain times. In this way, the cell can control what types of molecules are inside and outside of the cell, and that can affect the energy disparity or the energy a cell might have to work with. Just keep that little nugget of information in your head right now. That's going to become really important in the muscles and neuro part of this course. Different types of diffusion. Simple diffusion is totally passive. Molecules move right through the lipid bilayer in and out of cells. And this picture over to the left that I accidentally cut off showed that nicely, but I'll sort of redraw it. So you have lipid bilayer. I'm just going to draw squigglies in there for the phobic part. Okay, you have the inside and the outside of the cell. I typically mean that when I make these drawings, if I don't label it, the outside is usually the top and the inside is usually the bottom. So you have a molecule where you have them packed at a certain density on one side of the barrier, and you have the same molecule at a different density on the inside of that lipid bilayer. The density here is higher than the density here. So provided that they're capable of moving through the lipid bilayer, they will until, let's erase 1, 2, until they have roughly the same number of molecules on the inside as they have outside. This can happen in molecules of the same type or different types. We're talking about molecules of different types involves knowing stuff about their like electronegative pressure and complicated chemistry concepts that we don't need to know here. But simply think of it as the more solid stuff you have diffused on one side of the cell, if you have more on one side of the cell and less on the other side, as long as those things can just passively diffuse, they can get through the lipid bilayer, they will until the concentration of that stuff is equal. Facilitated diffusion is sort of like simple diffusion, except it requires a channel. No energy is spent during facilitated diffusion, but things are sort of selectively moved. So you have a channel, okay, of some kind that has a carrier-mediated facilitated diffusion. You have a receptor of one kind that has a place on it that's shaped exactly like what it wants to have come through. That'll open up to the environment, and as long as this fits, it's going to be allowed to pass through. Same thing in the other direction. It could also export molecules the same way, technically. If this doesn't fit, if you had a different molecule over here shaped like, oh wow, that's bad. Shaped like that, trying to get through, it wouldn't. It wouldn't fit into that spot right here, and therefore the channel just wouldn't do anything with it. It could also be a more sort of straight type of channel that can pass through based on other chemical interactions. Sometimes this channel is not specific to the molecule, but it's specific to a different molecule in your body that will merge to the molecule of interest and then bring it in, like a lock and key situation. A classic example of this is insulin and glucose getting into your body or getting into your cells. So you pound back a Coca-Cola. You now have a ton of sugar in your bloodstream. That sugar is great energy source for your cells. Your cells want that simple sugar, that monosaccharide, really badly. It's a great energy source. It breaks down very simply. The cell doesn't have to put in a bunch of energy to use it. It's great. But glucose is a fairly large molecule and can't just work its way through the cell membrane. You need a channel to bring it in. You only want to bring in as much as the cell needs. You don't want it to just come on through. So you want to have a channel that will let this in, but not just based on concentration pressures. So you make insulin, right, to bring down your blood sugar. What does that mean? Insulin will come in. Actually, let's pretend that glucose is this star molecule, and that's what you want to bring into the cell. And these little guys are insulin. What'll happen is insulin will bind to the glucose. Insulin can fit in the channel. And since it can fit in the channel, it can bring itself into the cell as well as the glucose. That's how the glucose will get into your cell. And then the insulin can get back out, no sweat. Osmosis refers to the movement of specifically water molecules. When we talk about diffusion, you're going to think of non-water molecules first. Osmosis refers to water specifically. The key statement to remember is water follows solids. Hopefully you learned that in introductory biology. Where you have concentration gradients, not only will molecules perhaps want to move, right, other molecules, but water molecules are going to want to move too. They're subject to the same types of concentration pressures. So let's pretend you have a solution where you have a bunch of a given molecule on one side of the cell membrane that has no real way of getting in or out, or was able to get in but can't get out. Okay? Since you've packed a lot of that molecule into the inside of the cell in this case, on either side really, but the example is inside. Now the concentration of water is lower. You have the same amount, but since you packed in more stuff, the ratio of stuff to water is different, right? There's more stuff than water, or at least there's more stuff compared to water than there was before. Water, being a molecule just like anything else, is going to want to equalize its concentration gradient. And so water is going to want to travel to wherever those solids are, so that the ratio of water to solids on one side of the membrane becomes the same as the other side of the membrane. Water, being hydrophilic, being the definition of hydrophilic, can interact with the surface of the cell membrane, but it can't get through because the inside's all phobic. So it needs its own special channels to get in. These are called aquaporins. Aquaporins are the channels along or through which osmosis happens with a cell. Active transport requires energy. So these three forms, no additional energy. This requires energy in the form of ATP. Remember, that's our basic energy currency of the cell. When you need to move molecules against their concentration gradient. So let's take this example for a moment. Let's pretend instead of moving molecules this way, because you have a lot out here and less in here, let's pretend that for whatever reason the cell wants to make lots more of it out here than in here. That's against the concentration gradient. You're moving molecules towards the direction in which there's already a lot of that molecule. There's no way to passively do that. Molecules are lazy. They're going to go wherever it's easiest to go. You have to burn energy to make them move across their concentration gradient. If you think of a dam, there's a lot of water on one side of the dam, and there's almost none on the other side of the dam. Water will flow automatically to the side with less water. It's a passive process. It's just moved by molecules and gravity and whatever. If you wanted to push water from the low end of the pool, from the low end of the river, up to the high end, you'd have to go down there, you'd have to scoop it up in buckets, go all the way to the top, dump it in, or install a pump and draw a tube and then run that pump to pull the water up against the gradient. You're inputting energy into that process. Similarly, your cell needs to input energy to move things in the opposite way that they would ordinarily go. Remember that the interior of cells and body fluids are based on water, but they're not pure water. So there's always this stuff around. You have water, but you also always have stuff. How densely packed that stuff is in the water is its tonicity. You have salts, you have sugars, you have fats, you have bigger proteins, you have whatever floating around in varying concentrations. The ability of a solution to change the shape of cells based on its osmotic properties, based on the difference in concentrations, refers to tonicity, in other words. We have hypotonic solutions, isotonic solutions, and hypertonic solutions. These are all relative terms that compare the solution on the outside of a cell to the inside of a cell. If you have less stuff outside of a cell than inside, it's referred to as hypotonic. Hypo is less or low. So the solution outside the cell has less stuff, less concentration, less osmotic pressure than the inside. Hypertonic, hyper means high or lots, is when you have lots of stuff outside the cell compared to the inside of the cell. I want to stress that this is a relative term. You have to have something to compare it with to use the terms hypo, iso, and hypertonic. You can't just say blood is hypertonic. That doesn't make any sense. Compared to what? You need to give another thing. When we're talking about cellular biology like this, we're always talking about the inside of a cell. But when we're talking about other systems, you're going to need to make that clear by context. So a hypertonic solution is where you have lots of stuff compared to the inside of the cell. And so it's osmotic pressure, the amount of force with which it wants to pull water through towards it, is higher. If those pressures are the same, it's isotonic. Water can move back and forth. Molecules can move back and forth, but slowly. And there's not really a whole lot of incentive, so to speak. There's no pressure pushing anything in any direction. Things more or less stay where they are. When you're given an IV solution, you are going to be given, typically, an isotonic solution. You come into the ER, you're super dehydrated, you had the flu, you're really dehydrated. They're going to start you on IV fluids. When they administer those fluids, they hope that administering that fluid doesn't kill you. If they administer a hypertonic solution, the solute in your blood is going to get high. It's going to suck fluid out of your cells and dehydrate you even more. Bad, right? If they administer a hypotonic solution, like if they just put regular old water in your bloodstream, there's going to become less stuff than the inside of your cells. The osmotic pressure, water follows solids, water is going to have a force of moving inside, and you risk exploding the cell. If you pump too much water into the cell, it's just going to burst. Also bad. So unless there's a really specific reason why you would want to administer a hypertonic or hypotonic solution, you will be given an isotonic solution. I know at one point in time they did. I don't know if they still do, but they did for a very long time. In cancer patients, some types of cancers, well, what the surgeon's going to do, let's pretend there's a lung here and there's a big nasty tumor here. The surgeon's going to go in and cut out the tumor and some space around it. Because remember, tumors spread and they metastasize, and it's very minimal, they grow bigger. And that means it has to be adding cells outside, on the outside of itself somewhere, somehow. When you go in to somebody with an obvious tumor, the tumor's obvious, you can see it, you can take it out, but you don't know if there's still some cells in the adjacent tissue that are cancerous, and so will just grow back on their own after the surgery. You don't want that. So they'll cut out that tumor plus a little extra. And what they did for quite some time is they would flush out this area also with pure water. Pure water has no solute in it. It is just H2O. It is the most hypotonic solution you can have. They would irrigate, spray water on, that area where they just cut out, and any adjacent exposed areas over which you might have dragged tumor tissue or the tumor tissue lingered over, in case there are yet still more cells left behind on that surface. The theory being the water bathing the outside of that cell is going to rush in to the cells on the surface and explode them. Whatever's there is just going to explode. They then suck out the irrigation so you don't just kill everything. But that was a way of helping control tumor metastasis and accidental, not incomplete removal of tumors at one point in time. So going back to active transport for a moment. Again, the only one that requires ATP. Everything else is some type of passive. Facilitated diffusion has a helper, but there's still no added energy, so it's still considered a type of passive transport. It's not active. There's a couple different kinds of active transport. Primary and secondary mainly, easily named. Primary active transport requires breaking down ATP directly. Is that hydrolysis or is that dehydration synthesis, turning ATP into ADP and a loose phosphate? It's hydrolysis. Hydro, water, lysis, breaking. The breaking part should tell you need to take an ATP and break it apart to release energy. So it's going to be the one with lysis in the name somewhere. So ATP is going to come along, excuse me, and it's going to react with a molecule on the inside of a transport protein and break into ADP and phosphate. When that happens, energy is released. That energy is used to open up a molecule, to open up a channel and push ions through. So the molecule started, the channel started, and remember, we're always doing this against a concentration gradient, moving things from low to high concentration instead of the other way around, which would be easy. So the channel's open to the inside, and you have stuff that can bind here. It's wide open. It'll just bounce around and touch there eventually, but you need to get it out. So you bring along an ATP, pop off the phosphate, release lots of energy, and that'll move the channel into a different direction. Also, at the same time, it will typically change the shape of where that protein was sitting in the first place. You see how it started off with a angle and then it turned round? That's going to help pop off the solute as well. So you change the shape of the channel so that it opens up to the other side. It changes the shape of the receptor on the channel so that it pops off the thing that was bound to it. And then when that energy dissipates and the ADP and the ATP are gone, or the ADP is gone, it moves back to its original form. One ATP comes along, pop, opens, moves, done. Secondary active transport involves one molecule moving along its concentration gradient to pull something else along with it. Sodium glucose channels is an example of this. So what you have is this type of pump, okay, that kicks sodium outside of the cell against its concentration gradient. That sodium is permitted to leak back in by special sodium glucose channels. When that sodium leaks in, it's able to bring a little bit of glucose along with it. So I told you about the insulin getting glucose in. That's the main way you get glucose into the cell. You get a little bit this way too, but it's not hardly anything. You really need insulin to survive. So what secondary really is this process plus another incoming process with facilitated diffusion, where you have one molecule that's coming in along its gradient and dragging something else along with it.